Hello, this is Professor Young, and we are ready to explore part two of the lymphatic system. Uh, we will be covering in this session the remainder of the structures or organs that are considered to be of the secondary lymphatic system, and then we will begin to go into exactly how the system, lymphatic system, works with the immune system to mount a defense against any kind of foreign bodies. And so with that, why don't we begin with the spleen? We left off with the last session under tonsils. Excuse me. We left off with the discussion of the tonsils, and now we're heading into the spleen. Of course, the spleen is located kind of inferior, yet dorsal to the stomach, and so it's kind of a weird shape or a weird placement. So it's underneath the stomach, but yet it's tucked kind of back behind the stomach, all at the same time. And within the spleen, we see two very distinct areas, which will become obvious on the next slide. We have a red pulp area, which is simply a concentration of red blood cells. And more importantly for our discussion of the lymphatic system, it's got what we refer to as a white pulp. And this is where the function of the lymphatic system is really going to take place, because this is where we find the lymphocytes and the macrophages. The production, or the function, I should say, of the spleen within the red pulp area, within infants, we see red blood cell production. This also acts as a graveyard area, again, for red blood cells. But more specifically, when we talk about the white pulp, it is a monitoring station for the antigens and any kind of immune response that may need to occur. So as you can tell, we've got dendritic cells, we've got the lymph nodes, we've got the spleen, we've got all of these various regions. We've got the tonsils that are going to be on guard for anything that may try to get into the body, so that the appropriate response can be mounted. The spleen is also going to, again, go ahead and conduct phagocytosis within the red blood cells. Again, that's going to happen within the red pulp. And then it's going to monitor for bacteria and for debris. And that's mainly going to be occurring within the white pulp. Here is the slide that I was referring to in lecture. You can see the areas of red pulp and white pulp much clearer with this slide, at least in this view. So right in this area, we have an area of red pulp. Right in this area, we have an area of red pulp. Again, in this area, we have an area of red pulp, and we have a little bit of an odd-long section here of red pulp. Everything else that you are looking at, and when I say everything else, I'm going to actually draw this out because I don't want any mistakes here. But basically, this entire area that I am outlining for you is all going to be white pulp. Now, notice I've left out some areas, specifically, I've left out kind of like in this area here, I've left out this area here, I've left out. What we see happening within those areas is kind of a mixture between, it's not really solidly red pulp, it's not really solidly white pulp, but it's kind of a mixture. And then peculiar as it is, we've got these areas right here. There's one there, There's one there. We have a little bit of a section of it down in here. And again, right in there. And what we see there are blood vessels. That's what we're saying. So this is what a typical spleen would look like if you were checking it out for histology. Now, let's start moving into some of the good stuff anyway, and that is the defense. We know that any good sports team has one of the necessities is an awesome defense. That's what allows, or that's what prevents any offense to progress or happen, which typically will prevent any scoring, unless you're in baseball, at which point the defense is actually in control and not the offense. But we're not going to get into any kind of sports theology now. But I want you to think of the immune system as being a defense team. And we have to have these linebackers in place ready to stop any kind of a rushing game that may happen. We need to protect the quarterback. The quarterback in this case is the body overall. And the linebackers are going to be there to stop anything that the offense may be trying to put together. The offense, in this case, is considered the bacteria or the viruses, and they want to move themselves throughout the body and score their points. And the way they're going to score those points is by infecting the body. And so these various types of cells are going to be there to prevent that. We have really two major groups of linebackers that we need to be mindful of. We have group one, which is going to be consisted of or make up the external barriers as well as our leukocytes and our macrophages, antimicrobial proteins, immune surveillance, your dendritic cells, cells of the tonsils, things like that, as well as inflammation and fever. The reason why we include inflammation and fever into this category of first-line defense, if you would, is because the job of inflammation and fever is to go ahead and kill off the bacteria. Again, external barriers such as skin, mucous membranes. Your skin, as you sit here living and breathing right here in front of your computer, you are being bombarded with bacteria. You're being bombarded possibly with viruses. You're being bombarded with things that are not natural to your body, but is trying to get into your body. And what is protecting you from that stuff is your skin. It's your mucous membranes. That is secreting mucus that's going to capture this stuff before it even enters into the body. I was mentioning in class I've done some work with tilapia and aquaculture, and within that work, one of the things that I would always have to make sure with students is that they would always wear gloves when handling the fish, because your hand will actually, by handling the fish, your skin removes this natural slime layer, which is really just mucus that is surrounding the fish. And when you do that, you remove its immune system. And so we've got something very similar to that concept. Now, both the external barriers and your leukocytes and macrophages and everything are not bacteria specific. They're not virus specific. They're not pathogen specific, if you would. These two groups tend to be more shotgun type of defense mechanisms. In other words, when a shotgun lands, it scatters and spreads. And that's exactly what these external barriers, as well as this broad spectrum group of defense mechanisms, really do. They ward off a whole range of pathogens, bacterial infections, viruses, so on and so forth. Whereas the immune system, specifically, right, the immune system is very stealth-like. It's very coordinated in its attack. And what we mean by that is every single immune cell, such as your T cells or your B cells within your body, will typically have already been exposed to a certain antigen, and it knows that when that antigen is within the body, the dendritic cells, let's say, send a signal over to the thymus, hey, gather your T cells that are already pre-programmed to deal with strain 2 of the influenza, let's just say, or strain B of influenza. It'll send those T cells out, and it's a very specific, targeted way of attacking the pathogen. In that case, there it would be influenza. I would suggest that here's for the leukocytes, we're going to talk about briefly with the neutrophils, but I would suggest going to your shared files and pulling the slide up, because what I cannot do on here is show video clips, but we have, at least right there, a really good little video clip that was found on YouTube of a neutrophil hunting down a bacterium. And that's exactly what we see neutrophils doing. Neutrophils are typically going to be located within the connective tissue, and it relies on two very different processes for getting rid of the bacteria. Phagocytosis is something that we have been through over and over again. I'm sure you are getting tired of hearing me talk about it, so therefore I'm not. Other than just to remember to say that the little video clip there is going to basically show a neutrophil doing phagocytosis on a bacterium that it is hunting down and chasing within the body. But then we've got this really, you need to think about this process. You've got this really cool system known as a killing zone that neutrophils will use. And it's basically a suicide mission that the neutrophils are going, because the killing zone actually not only kills very effectively the bacteria, but the killing zone also kills off the neutrophil. Now here's what happens, and I'm going to type some of this out, so you need to bear with me, because I can't type and talk at the same time very effectively. But you have these, we have structures within the body called lysosomes. And lysosomes are the garbage men, if you would, of the cells. They're there to clean up debris. And one of the ways that they do that is by releasing enzymes into the interstitial fluid. So the lysosomes are going to be releasing these enzymes into the interstitial fluid. But what ends up happening is that these enzymes are going to react with, if you would, they're going to react with oxygen, O2. And when they do that, when the enzymes react with the oxygen, it is going to form superoxidase or superoxide. Now, what is superoxide? Superoxide ions are typically free, or typically just free radicals. It is O2 with a dot and then that negative sign, which means that it has an unequal number of electrons. It has added electrons. They are unequal in number, and that is basically what a free radical is. And so that superoxide ion it is what it is. It's a free radical. What is amazing with this, and this is all happening within the tissues. All right, now your lysosomes have ruptured. They've released the enzymes. These enzymes are soaking up oxygen, which is then creating superoxidase. What happens from here? What happens from here to me is just amazing. The superoxide basically joins with hydrogen ions. The superoxide joins with hydrogen ions. Now remember, and because I can't do subscripts and superscripts on this, I'm not doing that here. There's probably a way I can do it, but at any rate, when we say hydrogen ions, right, what we've got to keep in mind here is that hydrogen and even oxygen is not just floating around the body with one oxygen or one hydrogen. We always find those natural gases. We always find them as diatomics, which basically means two of. So there's always two hydrogens or two oxygens floating around, whereas two chlorine molecules, if it's chlorine gas, is going to be floating. Two helium molecules if you're dealing with helium. So whatever it is, it's going to be always at least two of in the case of atmospheric particles. So that superoxide is going to join with the hydrogen ions, and what it's going to form is hydrogen peroxide, which is H2O2. And that hydrogen peroxide is also then going to produce. Now, hydrogen peroxide, I should mention, is toxic to the body. It's not friendly. Yes, we use hydrogen peroxide to clean out wounds, but it's also doing more than just ridding the body of the bacteria and dirt. It's also actually damaging good cells as well. But again, the plus outweighs the negative, and so we go ahead and we continue to use it. But along with that hydrogen peroxide, hypochlorite is also, or hypochlorite, hypochlorite is also formed. Both of these, hydrogen peroxide and hypochlorite, are both highly toxic to the body, and it forms this gaseous cloud surrounding the neutrophil, and that gaseous cloud becomes the killing zone. So that if anything enters into that killing zone, it dies, and then the neutrophil is there to basically go ahead and suck it up and break it down and do its phagocytosis, but eventually it'll end up killing the neutrophil as well. And so everything dies with this. This is the leading cause of rheumatoid arthritis. Uncontrollable lysosome splitting open, releasing the enzymes, mixing with oxygen within the interstitial fluid, forming the superoxide joining forces with the hydrogen ions, forming hydrogen peroxide and hypochlorite, and then basically having these whirling masses of killing zones surrounding by these neutrophils going through the joints within the hand, killing off the healthy tissue, killing off the healthy cartilage, and killing off the joints themselves, causing swelling and pain and stiffness. And that's rheumatoid arthritis right there. Fascinating stuff when we start looking at this. Fascinating stuff. Highly effective. Highly effective. A little bit more on leukocytes. Of course, we have the eosinophils. We already know from past discussions dealing with blood that eosinophils are going to protect against the parasites. They're also going to be activated for allergens and parasitic infections. Basophils are there again to support the leukocytes. Histamine is a vasodilator, helps to speed up blood flow. Heparin is a blood thinner, which is going to prevent blood clots. And so that's what basophils are going to be responsible for. And of course, we have lymphocytes and monocytes. We don't have to say too much about that. Lymphocytes are your natural killers. Attack and destroy, attack, find, and destroy. And then you have your monocytes there as well. What I do want to kind of touch on are three other rather cool ways in which we can have this broad spectrum of defense within the immune system. Interferons are basically released into the infected cell, and they do that. Let me see if I can draw this picture for you. I don't think. Hold on one second. Yeah, it's going to be a horrible picture. These interferons I'm trying to think of the best way to do this. Here's our cell. It's a bacteria cell, or it's a virus. So let's say viruses. This is a viral cell. All right, now, sitting on top of this viral cell is this interferon. All right, now this interferon is coming over from an infected cell. And what it's going to do is, let's scratch that. Hold on one second. I'm getting my natural killers confused with with everything else. Hold on one second. This here is a normal cell, the red. The blue is the interferon. It's going to enter into the cell, and when it does that, it's going to release a protein within that cell that's going to basically be able to block any kind of viral replication. We talked about in lecture the fact that viruses are not living. And the chief reason why they are not living is because they cannot replicate on their own. They have no means of providing reproduction. They have to rely on the host. No doubts about it. And so what the virus will do is the virus will basically land on the cell, drill a hole into the host cell, inject its DNA or RNA into that host cell, and that DNA and or RNA basically becomes infused within the host cell's DNA. And when the cell replicates and divides, so does the virus. And then the number of viruses becomes so overwhelming within that cell that the cell wall ruptures, or the cell membrane ruptures, releasing all those new viruses throughout the body. Interferons block that, which has major pharmaceutical and clinical applications. I mean, interferons do what pharmaceutical companies have been trying to replicate for 10, 20, 30, 40, 50, 60 years, if not longer. And so it's amazing that all they got to do is really look at this process and say, all right, if we can block the receptors that are allowing the viruses to attack or attach to, then we can stop the virus. And that's what a lot of your antiviral drugs aim at doing. Keep in mind, there has never, and I will say this, there has never, up until April 2011, there has never been a virus cured. All we simply do is seem to control and maintain in other words, we've created therapies to deal with viruses, but we've never cured a virus. And that is so pivotal. That is so pivotal in the sense that we've never been able to cure a virus. And our body doesn't really have an effective way of curing it either, other than these interferons, which do a pretty darn good job and are more successful typically than drugs that we encounter. To go along with that, we have the complement system. No, it's not going around and saying, it's not giving compliments all over the place. But when we say complement in this sense, we're simply meaning that it's going to work alongside other immune reactions to strengthen them. And that's basically, and it's going to do that chiefly through inflammation. All right, now going back to what I was constantly getting confused earlier. Here we have natural killer cells. We find natural killer cells besides within the lymphatic system. We also find them within red blood cells. We find them within the plasma within the blood. And they're typically about 5 to 10% of those white blood cells are considered to be natural killers cells. And again, this is your lymphocytes typically here. We can also find them within the spleen. We also find them within red bone marrow. We also find them within lymph nodes. But what these natural killer cells do is attack, destroy, annihilate any kind of abnormal cell. And it does that by releasing a toxin known as perforin. Now, when you see the word and hear the word perforin, you should be thinking perforated, which means to tear or to rip. And that's exactly what this structure does, this perforin does, which is a protein. It basically makes a hole into the membrane of the virus or the bacterium. Now, I'm going to go back to what I was trying to do on the previous slide. Here's that bacterial cell. Here is our perforin, which is an enzyme. What it does, again, is going to rip a hole, a channel, if you would, down into the membrane. And so it's going to have a little channel here. I'm going to try to draw this as best as I can. No laughing, please. So here's our channel going down into the cell. And then what will happen is the enzyme perforin will go ahead and release granzymes. So out of this enzyme, all right. Again, perforin has been released. It's going to go ahead and drill a hole through the membrane, which is going to then allow these granzymes to come in, enter down into there, and once the granzymes are in the cell, they basically force that cell to go through apoptosis. There's that word again, programmed cell death. And that's what happens. These guys basically self-destruct, if it was, implode. The bacterium dies, and then you have phagocytosis that basically goes ahead and occurs. So this ranks right up here to me. Natural killers ranks right up there with the killing zones. It's a fascinating mechanism by which we have to control bacteria that are entering into the body. And then last but not least, we have inflammation and fever. And this is pretty straightforward. I'm not going to spend hardly any time on this with you guys other than there it is. We know it happens and it's for the best that it does happen. With that, I'm going to leave you. Please, as I always say, review this material. If you have any questions, shoot me an email as soon as you think one's there, no matter how minor or major that question may be. If you don't want to shoot me an email, jot it down. Ask me during the next class. I will always give you that time. You know that. And until we meet again in lecture, happy studying. And this is Professor Young saying good night, good day, good life, whatever it may be for you right now at this point in time.